Hi everyone, welcome back to Biology 101. This is Amrita and today we have linkage and recombination, a very fun topic to learn. Okay, then why I wasn't posting for 9 to 10 days because I was not well. There were some other issues and then I had my second round counseling going on as well. It's still going on. I'll try to post regularly from today. Okay, so linkage and recombination. We were learning the last time we saw was the theory of inheritance, right? Chromosomal theory of inheritance. Now here Morgan carried out several dihybrid crosses in Drosophila. Now, Morgan started experimenting in Drosophila, right? Drosophila melanogaster, that is the flies. Now Morgan carried out several dihybrid crosses here to study the genes that were sex linked. The crosses were similar to dihybrid crosses which was done by Mendel, right? Mendel did the crosses in peas, and here in Drosophila it was done by Morgan. Now for example, Morgan hybridized yellow body, that is yellow body, and white eye, okay, white eye, to brown bodied red eye, to brown body and red eye, male, here female and this is male. So when he intercrossed their F1 progeny, he observed that two genes did not segregate independently. We learned the process, we learned that independent segregation and all, right? But here what he found, that two genes did not segregate independently of each other, and the F2 ratio deviated. In Mendel, what did Mendel say? Mendel said that F2 ratio gives 9:3:3:1, right? But what Morgan found was F2 ratio deviated very significantly from this ratio expected when the two genes are independent. This is expected only when the two genes segregate, when they segregate independently. Okay, but what Morgan found was it is not giving 9:3:3:1 because he's saying that these two genes did not segregate independently. They did not segregate independently. So Morgan and his group knew that the genes were located on the X chromosome, okay, and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, that is one chromosome, we have one chromosome, in this two genes are located on the same chromosome, the proportion of parental gene combinations were much higher than non-parental type. See, let's say this is parental type, two genes are present in one chromosome, on the same chromosome, right? In the progeny also we could see similar to this, that is parental means same type, non-parental means different. So Morgan is saying that when a chromosome has two genes in the same chromosome, the proportion of parental combinations will be higher, that is greater than the non-parental ones. I think you're getting it, because when they are in the same chromosome, they will not segregate and they will not recombine. When they recombine, only non-parental will come. When they don't recombine, it will be parental, that is same as the parents. That is what Morgan is trying to tell. Morgan attributed this to the physical association or linkage of the two genes. That is, these both are together, right? So how will they separate? That is physical association or linkage, and thus he coined this as linkage. Morgan only coined as linkage, okay? This word was given by linkage, Morgan. Linkage word was given by Morgan, very important, to describe the physical association of genes on a chromosome, and the term recombination. Recombination also was given by Morgan. Okay, very important. Recombination and linkage, both the words were termed by Morgan. What is recombination? That is, from here one gene goes with another gene, that is segregates independently, then it is recombination. Otherwise it is they don't recombine. See, recombinations means again they combine, right? But that is when they recombine they form non-parental. When they don't recombine they are parental. That is same, they are same as the parents. Morgan and his group also found that when genes were grouped on the same chromosomes, some genes were very tightly linked, showing very low recombination. That's what, when they are in the same gene, no, they are very tightly linked. When they are together they are very close, right? So they don't show much recombination, show very low recombination, okay? While others loosely packed, when they are loosely like let's say here one gene is there, here one gene is there, and in the next chromosome here one gene is there and here one gene is there, then it will be more easier for these both genes to recombine, right? Here it will be easy to recombine, here it will be difficult because these both are strongly linked together. So here there will be more non-parental, here there will be less non-parental and more parental. So when Morgan did cross in the Drosophila, what he found was 1.3% recombination, only this much recombination in white and yellow, okay, but 37.2% recombination in white and miniature wing. So these were the differences he found. His student Alfred Stutwent used the frequency of recombination between gene pairs of the same chromosome as a measure of distance between genes and mapped their positions. Let me explain. What his student did was he used the frequency of recombination, that is how many it's where and all it's recombining, when are they recombining, are they recombining when they are close or far. He was seeing between the gene pairs on the same chromosome as a measure of distance between genes and map their positions. He told if the genes are close, then their recombination also will be very less. When they are far, there will be more recombination. That is, he told the distance will be equivalent to the recombination ratio. That is, they mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing, of which the genomes was done in the case of Human Genome Sequence project. The Human Genome Sequence, how it is done, how this mapping was done and all, we'll be learning the next chapter, molecular basis. So that is what basically linkage and recombination is. This was mainly given by Morgan.